So, good evening, distinguished members of the society and invited guests, professors, emeritus professors, founders, respected members. The webinar this evening will start now, and I will invite the moderator, Ms. Evelyn Alanas, to kick us off. The presenter is also here with us. So over to you, Ms. Evelyn. Hello, Miss Evelyn, can you hear me? Oh. Hello, Miss Evelyn.
I can hear you now.
Please, we are waiting for you to start the moderation.
Yes, sir. Can you hear me?
Yes.
Yes. Am I audible now, sir?
Yes.
Okay. Thank you, sir. Good evening, everyone, and thank you, sir, for the opportunity to moderate tonight's session. I'm Evelyn Ananias, and we welcome everyone to this series of webinar. Today we'll be looking at preclinical implications of the clinical implications of preclinical pain research, and we already have a distinguished speaker who is ready to speak to us today, Dr. Ahmed. Before we start, I would love to, before we invite him rather to take the floor, I would be reading his citation tonight. Just a minute, please. Is a senior lecturer in the Department of Human Physiology at Ahmadu Bello University, ABU, Zaria, where he earned his BSc in 2008, his MSc in 2012, and PhD in 2017. He later completed a postdoctoral fellowship in anesthesiology and critical care at the Stellenbosch University, South Africa, under Professor Sion Chetty, during which he helped strengthen interdisciplinary pain research between departments. As a neurophysiologist, Dr. Issa's research centers on pain physiology, particularly the chronobiology of pain and its relationship with weather and climate. He also investigates neuropathic pain, the effects of neurodegenerative diseases on pain processing, and the analgesic potential of cannabis. He is passionate about advancing translational pain research and has collaborated widely across scientific disciplines. During his postdoc, Dr. Issa served as national coordinator for the Pain Out South Africa Network, a Pfizer-funded project in partnership with German institutions. He is actively involved in science outreach through Trend and has participated in IBRO-sponsored neuroscience training in Tanzania, Lagos, and Morocco. His commitment to science communication was further shaped by the African Science Literacy Network. And being an avid reader and traveler, Dr. Issa also practices karate. And through his mentorship, research, and public engagement, he aims to inspire the next generation of scientists and make science accessible to all. Ladies and gentlemen, please join me in welcoming Dr. Issa as he takes the floor. You're welcome, sir.
Thank you very much. I'm grateful. Can I go ahead, please?
Yes, sir. You can go ahead.
Thank you very much.
Dr. Issa, if it is possible to put on your video because we are recording, that would be great. But if not, no problem.
Okay. I don't know if you can see me now.
We can see you.
All right. I would like to say good evening, President, professors present, distinguished members of the society, ladies and gentlemen. Issa Ahmed Sharif from Department of Human Physiology, ABU Zaria, and I'll be presenting on the clinical applications of preclinical pain research. I would like to take us through this couple of topics: why preclinical pain research matters, the translational pipeline from bench to bedside, the models, mechanisms, and the molecules or biomolecules involved in pain. Key preclinical pain models, just a couple of them, a sample. And then some case studies of successful and failed translation to clinical practice, some of the challenges and translational gaps, and future directions and clinical opportunities. So to begin with, pain affects about 1.5 billion people globally, and these account for about approximately 150 to 20% of the population. It is one of the major reasons, if it is not the main reason or the only reason why people seek medical attention. Because if they don't feel pain, they don't actually go to seek for this help or assistance medically. Considering the fact that it affects so many people, pain is still poorly managed globally. And because of that, according to the United States, it forms an economic burden of about $635 billion, which is equivalent to about 800 trillion naira. If we are to convert it, that's the equivalent. And according to the statistics from Nigeria, we have that about 30% of people live with chronic pain disorders. However, in about 30 years, excluding the drug that deals with calcitonin gene-related peptide, the CGRP, there have been no discovery of any new mechanism associated with pain in the last 30 years, except for this. It has including even anticonvulsants usage, which has been repurposed for the management of pain. So basically, there have been this 90% failure rate in translation of preclinical pain research into clinical use. And it means we have about 10% attrition rates. And this is one of the highest, if not the highest, failure rates that is observed generally amongst other medical conditions. So this basically means that there's a very large gap to be filled. The fact that about 20% of the population actually experience pain, either chronic pain in their various forms, from low back pain, which is the major contributory factor to the pain that people feel, fibromyalgia, and the rest of them. So it means that despite these low success rates, if there's a minimal improvement or breakthrough, it will benefit hundreds of millions of people, despite the low success rates of the clinical pain research. So basically, it remains the primary tool for which we can investigate pain mechanisms and validating therapeutic routes. And most analgesic failures usually occur when you have this translation from animal models to human models. That's usually around the phase 2. And then pain is also a subjective experience that involves quite a number of things. It's biopsychosocial, and it's not basically nociceptive. I think we'll probably talk about the difference between pain and nociception. And whether we like it or not, the researches or the insights we've gotten from clinical pain research basically has been the foundation for drugs ranging from opioids to NSAIDs, paracetamol, aspirin to anticonvulsant that has been proposed for neuropathic pain. So it still serves its purpose. So we'll begin by what is preclinical pain research. According to the National Cancer Institute, preclinical pain research is simply defined as research using animals to find out if a drug, procedure, or treatment is likely to be useful. So basically, it means the use of non-human species or subjects that could include in vivo experiments, in vitro experiments, whole animals, even tissues, and even in some cases, models like rodents and even Drosophila in recent times to carry out preclinical testing to find out whether there's any potential in analgesic biomolecule. So the translation basically from the National Center for Advancing Translational Science is the process of turning observations in the laboratory, clinic, community into interventions that improve the health of individuals and populations, from diagnostics, therapeutics, to medical procedures and behavioral interventions. So basically, the first stage in trying to find out drugs that can be used clinically is to start from the preclinical stage, where experiments are carried out in non-human. So the first thing is done in cells, either in cells or rodent, large animals, and computer simulations. And then based on the insights that are gotten from there, if there are any potentials, there's any interaction with the receptors, and then you find these beneficial effects, then it is now translated into the clinical stage where the human studies are done. So in phase 1 is usually done on healthy individuals, and then in this case, they're trying to find out if it is safe. And then in phase 2, you're now actually exposed to the subjects who actually have pain syndromes. And then you find out whether it actually works. Probably if you have a target receptor and then you block those receptors, does it actually relieve the pain? And then phase 3 is to have a much larger population from 300 and above, where you now actually see what effect it has, and then also monitor the safety profile. And eventually from there, it moves on to it being available in the market, where real life data is still continuous, and then approval moves. So to develop an analgesic that is going to be useful to patients, basically, as I just mentioned, you start with discovering the signal. Probably you have a target, you have a receptor that probably is implicated in pain. And then from there, we go to animal testing. If you block those receptors, does it relieve pain? Does it lead to pain relief in these animal models? And then thereafter, you have where you refine this molecule, making it safe, stable, increase the bioavailability, and then the efficacy. And then subsequently, you have the phase 1 to 3 trials in humans to test for safety and effectiveness as well as efficacy. And then the last part is where this analgesic is actually approved, and then patients can then find them at the pharmacy. So the average developmental time for you to have an analgesic from the time of discovery of the pain signal or receptors where it can actually act to the time where it's available for clinical use is about 10 to 15 years. So it means if in this year, now in 2026, we discover a target molecule that can be modulated to relieve pain, the earliest time we are likely to see that is probably in about 2036, and then probably 2040 something thereafter. And the average cost for development of an analgesic, or basically drugs generally, is about $1 to $2 billion. This is quite a lot of money. And combined with all these, you have a success rate of just about 10%. It's one of the lowest amongst these medical conditions. So what are the models, preclinical pain models, and their clinical relevance? So these models or experiments that are carried out in animals actually gives an insight into humans. We will definitely not begin to do our research in humans, especially in pain. We'll start with animals. And as I said earlier, apart from these rodent pain models, we are beginning to develop Drosophila models so as well that mimics some of these. So and they also have their own advantages too. So just a couple of them here. You have different pain conditions, and then some of them are relevant to these animal models are relevant to clinical pain that humans experience. So you have inflammatory pain where you have central sensitization of the pain pathways generally, and you have the implications of inflammatory biomarkers, basically the inflammatory soup. So one of the pain models in animals that is used is the complete foods adjuvant, which generally mimics inflammatory pain of the chronic origin, while the carrageenan injection has a shorter lifespan. So the clinical correlate is basically it mimics rheumatoid arthritis or post-surgical pain, which is associated with inflammatory biomarker. So you have drugs that have been developed from these, the NSAIDs, the COX-2 inhibitors, and then the tumor necrosis factor and interleukin-6 blockers. Then you have the neuropathic pain. You have the relevant models that are used here, the spared nerve injury, where some of the nerves are actually ligated and then others are spared, and that one becomes injured and it begins to carry out pain signals. So generally, we know that the neuropathic pain is associated with impaired somatosensory pathway or the nerves generally. So it's like having an electricity and you have a spark in between, and therefore it keeps sending ectopic signals of pain. So it could either be the chronic constriction injury, or you also have the streptozotocin diabetes model, which sort of induces diabetic neuropathy. In addition to this, you also have the chemotherapy-induced model too as well, where chemotherapy drugs can actually induce neuropathy. So it correlates clinically with peripheral neuropathy and post-hepatic neuralgia and CIPN. So the drugs that have been developed for this is gabapentinoids, duloxetine, which is basically the gold standard for the management of neuropathic pain, and then sodium channel blockers. Then you also have, for the modeling of cancer pain, we have the bone tumor implants, where implants or cancer cells are implanted close to the bone. And these basically, the clinical correlate is bone metastasis and breakthrough cancer pain. Why drugs have been developed from this is biphosphonates and the nerve growth factor inhibitors, as well as opioids. And then lastly, you have the migraine or headache models. So in this case, you have nitroglycerin, one of the few ones that mimics headaches found in human beings, causing vasodilation, and is also a strong inflammatory mediator. Drugs that have been developed from these triptans, CGRP, and monoclonal antibodies, which we'll talk about, and the GPAs. So basically, once you have these models that mimic clinical resemblance in humans or pain conditions in humans, then you now have outcome measures where you use the von Frey-Hairs that helps to measure mechanical odynia. That's expression of pain to non-noxious stimulus. You have the Hargreaves test that helps to measure the thermal hyperalgesia, and you have Rotarod, which helps to measure motor functions, as well as the Grimes scales that helps to measure the affective component. So these are new aspects to pain because of the complex nature of pain, not just being a reflex thing. Yeah, because one of the major problems we have with pain, especially with these outcomes, is that we measure reflex outcomes. So you put, after having this model, you put the animal on probably a hot plate, and then you try to measure thermal hyperalgesia. It takes the latency for the animal to react to this thermal noxious stimulus. So basically, it's nociception we measure when we are talking about animals. So basically, the physiological response to pain. However, pain is much more complex than that. It has affective components, autonomic components too as well, anxiety, depression associated with it. So these are some of the drawbacks of the preclinical models that we have, as well as motor functions. So the Grimes scales as well as the affective components are the ones that are being introduced, such that if you measure for pain responses, you also try to measure for the other components so that we can have a little bit more holistic measurement of pain. So we'll begin with the case study of some of the analgesics that have been developed from preclinical pain research. So we'll start with CGRP. And this has taken quite a number of years with the identification that CGRP are quite associated with migraine attacks because of their inflammatory effects, and they are basically found in trigeminal nerve. So, and they're elevated when you have migraine attacks. Thereafter, work has been done from 1985 to 1990s, where work was done in rodents, and then they moved on to develop a biomolecule, the first CGRP receptor antagonist, to show the efficacy, which was also successful. And in 2018, it was the first one was approved for a reduction in migraine, which is migraine prevention, followed by its successors. And then in 2020, 2021, 2023, we have the GPANs, GPANs, GPANs have been approved for acute treatment, and this completed the phase from bench to bedside. So basically, it has shown how preclinical pain research has been successful in getting to this point, and it has predicted the clinical benefits that we can get from blocking CGRP using antibodies also. So we also have gabapentinoids, which is another success story, which was originally developed for epilepsy, I think in 1993, and then subsequently it was repurposed for pain in 1996. So first, it was used in neuropathic pain using the chronic constitution injury model, which showed allodynia, mechanical allodynia, as well as the thermal hyperalgesia. And then subsequently, it was shown that you have increased expression of the alpha-2 delta calcium channels in neuropathic pain. And gabapentinoids tend to block this channel. So while studies were carried out in animals, they began to observe that they had side effects with drowsiness and dizziness in larger doses. And this was later seen when human trials began. So gabapentinoids was approved for gabapentin was approved for neuropathic pain, and then subsequently we had pregabalin, Lyrica, and they also observed that the same side effect seen in preclinical models as the animals was also seen in the humans. So that basically showed that the preclinical pain models was successful. It successfully predicted what will have been observed even in humans too as well, with good success. So even the dose was also that way. Now to the mixed results where these animal models did not really match human studies. So as I said, sorry? Okay. So we also have the nerve growth factor inhibitor, which is the first case study where animals, the animals models, they showed successful reduction in arthritis pain. However, by the time they got to human phase, they also observed that there was also reduced joint pain, which was very promising. However, they began to discover that it wasn't and actually increased exacerbated the joint destruction. And therefore, this study had to stop. Although it has been approved with certain regulations that if one has chronic osteoarthritis and has not received any benefit from all the other drugs, then it can now be used. So it's under restriction, or it has been given approval based on its on this criteria. And then you also have the nerve, that's the sodium voted calcium channel 1.7, which basically has been shown one of the ones that we saw genetical mutation in people, that when it was mutated, you had a loss of function. Basically, this led to reduction in expression of pain. And so they began to work on blocking this particular channel. However, in humans, it worked, but in animals, it worked, but in humans, it did not work. And later they discovered that the mutation or the blocking of the nerve 1.7 channel, or the sodium voltage sodium channel, actually led to an upregulation of the opioid system in animal study, which was not the same case in humans, because the blockage was actually acute rather than chronic, and it did not lead to the beneficial effect. Therefore, it was not successful. So you also have the nerve growth factor, which acts through which acts through the TRK receptors, that's the tyrosine kinase receptors A. And so it has been shown to be a strong mediator of pain sensitization in inflamed tissues in rodents. Antibodies of nerve growth factors were also given, and then it mimicked thermal and mechanical hypersensitivity. So preclinical studies actually showed that it was also beneficial. However, when it got into the human face, we began to observe certain safety concerns, leading to rapid progression of the osteoarthritis emergence. So I mentioned earlier that it has restrictions. So this is a repetition of what I mentioned earlier. So what are the problems with preclinical pain research and the challenges? Why do we have these mixed results? Why do we have just 90% success rate? Why is it there's only 10% that we have the chance of being successful? One of the major reasons is that we still have species differences. What I would like to know, despite the fact that we have homologs and similarity in conservation of the pain pathways between rodents and humans, there are still differences, especially with regards to pharmacokinetics, especially with the cyclotron chrome P450, which helps in metabolism. It's much higher in rodents than in humans. You also have the 10 times higher opioid receptors in rodents than in humans. And this will obviously influence the results that we get from rodents. We also have the problem of sex or biological variability, or sex bias, so to speak. So about 80%, that's before 2015, about 80% of preclinical pain research are usually done in male rodents. And we know generally from studies and health seeking behavior that the female population actually expresses pain more than the males. This is open to debate, even though some people believe that it's because of the subjective nature of the assessment of pain. People feel that if you assess pain objectively, you will likely see a 50-50 expression of pain, because of the men may refuse to classify their pain as being very high because of the cultural exposure that people have. So most of the studies are usually done in males, while females actually show more pain. And studies have also shown that there are certain differences in the processing of pain in females than in males. So since 2015, the NIH has proposed that when studies in pain are going to be done, there should be a balance. There should be a comparison between males and females. I think this was what motivated me to carry out my PhD study in only females to represent the population that has been segregated scientifically, so to speak. Then there's this outcome mismatch between animals and humans. So generally, pain responses for humans are usually self-reported, which is the gold standard for reporting of pain using the numeric rating scale. So but for our animals, you actually have this reflex-based testing like paw withdrawal. When you apply a noxious stimulus, you try to assess using the latency, how much time does it take for the animal to actually respond to this pain. And then therefore we miss the affective component and the emotional component of pain, and how it affects pain, how it affects sleep, and the rest of them. So these are some of the things that prevents effective translation from preclinical pain research to clinical experience. You also have the difference between acute versus chronic modeling, because of most of the preclinical pain research lasts between 1 to 8 weeks, while you have chronic pain basically lasting beyond 3 months. So this mismatch actually leads to ineffective translation from preclinical pain to clinical experience. Additionally, you also have comorbidities, as I mentioned earlier. Pain patients, about 30 to 50% of people that experience chronic pain also experience anxiety, depression, sleep disorders, and these are all basically absent in animal models. However, suggestions have been made that while carrying out animal studies and you assess pain, you should also go ahead to assess anxiety and depression in them. Even though, yes, that is good. However, it still leads to having a battery of tests from pain to anxiety and then to depression. But that will significantly improve our validity of translation from preclinical models to clinical practice. And then the last thing is dose extrapolation. So a dose that for a 200 gram rat does not linearly scale up to a 70 kg human, because metabolic rates or even the enzymes are actually different in rodents than in humans. So these are some of the few things that affect how we translate from animal study. So despite all this, the future basically is one to actually use human cell models where you have IPCS from inducible pluripotent stem cells from people who are actually experiencing pain, and then this pain will be induced so that they express themselves, and then you carry out research on cells from individuals that actually have pain experience. The other thing is the organ on a chip. This is a form of simulation of the human nerve and spinal cord in like a chip where experiments actually be done as if it's a living thing. Then you also have the use of AI. AI is the big thing now that happens. There's hardly anything that goes on without the input of AI. Basically, it has come to stay. There's nothing we can really do about it. So we otherwise just embrace it and then use it to improve our knowledge. So AI can be built such that it can analyze millions of data points from actual patients that experience pain, and then these can help to even predict results from drugs. And it can also lead to personalized pain treatment. Then you also have the gene therapy, where you can actually give certain, I think there's this virus bound technology that actually turns on either opioids, depending on what you want, vectors. So you can either turn on opioid receptors to reduce pain or block receptors, as the case may be. So this is gene therapy. Then the testing that we have, if you can have testing that is homologous or similar between preclinical pain research, that's the animal models as well as humans, and these can actually be able to equate to themselves instead of having things that are obviously different. And then finally, having sex-inclusive research, which is basically what the NIH has recommended since 2015, for us to have researches that involve males and females, and then compare them so that we can understand the pain mechanisms. As I mentioned earlier, studies have shown that there are actually mechanisms that differ between males and females. So in conclusion, basically, despite the poor success rate being 90% failure, preclinical pain research is still essential. It's not just the first step, it's the fundamental for you looking for any painkiller or analgesic before you begin to start human trials. So it's a fundamental step. And with the experience of CGRP and gabapentin, they have shown how animal models have actually predicted the mechanism as well as translation, and how it has worked beautifully in humans. It shows that it is highly relevant. And despite the failure, it still reveals that of especially the sodium voltage sodium channel, that yeah, despite the failure, we still actually had some knowledge from it, telling us that the blockage of this ion channel led to upregulation of the opioid receptor, and this helped to understand our management of pain. And one of the biggest challenges, as I mentioned, is species differences, exclusion of female animals, and then the short experimental timelines. So these are just a couple of things that do affect. And the experiments in preclinical pain research all still forms a basis for even management, such that I think there are some school of thought who believe that even the management of pain now should be mechanistic, so that you can include you manage pain based on how you understand the mechanism in which the pain the person has. So it's mechanism based rather than just giving a blanket analgesic or painkiller to people. And probably that will have also led to the school of thought that believe that painkillers of that act through different pathways can be given such that it will benefit the patient, as the case may be. So new tools that involve human cell models, AI, gene therapy, are things that can help to bridge the gap between animal and pain research. So these are just a couple of things. So even though preclinical pain research has some drawbacks, if we'll sit down, look at it, in fact, that's one of the benefits, that we actually know what the problem is and we improve on these models, then you are likely to get better output. So better animal models plus more human-like testing will actually lead to reduced drug and better pain relief for the patients. So this is just the repetition summary. So I would like to end with progress is impossible without change. And those who cannot change their minds cannot change anything. This is by George Bernard Shaw, and basically means that we know that preclinical pain research is important. We know it has drawbacks. And if we sit down, modify, improve upon it, I think we'll have better success rates in translating from preclinical pain research into the clinical application. So thank you for listening. Thank you very much.
Oh, wow. Thank you very much, sir. Thank you, Dr. Issa, for that comprehensive and very insightful session. I want to believe so many of us have picked 1 or 2 points, just like I did myself. We really appreciate your time and for taking us through this. I mean, it was in depth, and I believe that it captured everything that we needed to know about the transitional landscape of pain researches. And particularly, I think what stood out for me was when you emphasized on the need to have sex-based modeling for those of us that do animal researches with rodents. So thank you very much. With this, I would like to open the floor to questions from the audience and our participants. So you can unmute, or you just drop your questions in the chat box and I'll read them out for Dr. Issa. Thank you. Any questions? Yes.
Okay. Let
you.
me open the floor by thanking
Do we
Dr.
have any
Issa
more
for.
questions?
Hello, can you hear me? Hello.
Yes, we can hear you, sir. Professor Shoyawo's hand is raised, sir.
Okay, let Mama go on.
You can go ahead, sir. Yes, sir. Professor, you can go on.
Yeah, I just want to. Good afternoon, everybody. Or good evening.
Good evening, ma.
Good evening. Good evening. Good evening,
ma.
Yeah. I want to congratulate Dr. Sherif Issa on this beautiful presentation, and also congratulate him on his journey, because I saw that his pathway towards pain management and pain research actually started from basic sciences, and then I heard him talking about anesthesia and intensive care. So I have 2 questions. I want to know whether he works within an interdisciplinary team to make excuse me, his products and also his research meaningful to the use of our society, because there's a lot of issues with pain in Nigeria, in that a lot of people still don't understand what it is. But I know that in neuroscience, progress is being made in terms of interdisciplinary and multidisciplinary research. So it would be nice for centers that are carrying out a lot of preclinical research to also work with those who are working in the clinical aspects. And I'm happy that SSPN provides that platform. And that is why SSPN is unique in the country. The second aspect is that you mentioned the pain out program of the Federal Ministry of Health, which was run when we had opioid analgesics was being reintroduced into the country. I just want to know which area your research was on in that aspect. Thank you very much. And please keep on going and encourage others to join. I'm happy that I've seen a lot of names, physiotherapy, anesthetists, other clinicians, and a lot of basic scientists on the platform. I don't know the names, but I'm sure that the president is working hard to keep this group viable and going. Thank you very much, everybody.
Thank you very much, ma'am. Do I go ahead to respond to those questions, or do I just take them down?
I think you can go ahead while we wait for other questions. Thank you, sir.
Okay. Thank you very much, Prof, for your insights and feedback. Working in an interdisciplinary or multidisciplinary team, I know that during my postdoc, it was something that was interesting to find, because I was asked to attend a pain clinic where there was a pharmacologist, a medical doctor, anesthesiologist, I think a surgeon, and then a psychologist. And then I was with he was a medical doctor as well as a pharmacologist. So I was with him, so I was shadowing him. And then we had very interesting sessions together. That was where I saw this multidisciplinary team managing pain. And I know that, to the best of my knowledge, this was something that Prof. Onwaso, who also mentored me as well, was trying to do at the time. But coming back home, that made me to focus on translational pain, which I've been trying to do, even though not to the best of my ability. I wish I could still do more. So I've been collaborating with people in anesthesia department, and then even in oncology, where we are working on cancer pain. I think the next one that I've been working on for a while, and I've been very slow about it, is trying to see how to improve pain management in sickle cell patients. So somewhat I've been interacting with the clinicians in trying to see how it can be beneficial, or how we can improve pain management in patients. And the second question is about the pain out. So the pain out I was involved in was the one in South Africa, not the one in Nigeria.
Okay.
Yes, and
Okay.
I think it was just to improve pain management in post-operative pain, which we've been able to work together. We have 10 hospitals. We collected data, and then we tried to implement 4-element bundles. Very simple things, educating the patients, using round-the-clock analgesia, and then the rest of them, about 4 of them. And we saw how those 4 tend to improve pain. We had 3 stages: baseline, then intervention, and then subsequently we took another data. And these very simple elements improved pain management across the hospitals. So the plan was to probably come back home and then replicate something similar, but I have not been able to do that.
Thank you very much. Well,
Yes.
it shows you that we can do things properly in the country, because it's the same kind of grant that was also given to us here, and we did it using the personnel that we have. So, and what I want to bring it out is that it shows you that there's a niche for the preclinical researchers and also clinical researchers, even within our system. Maybe you should find your way to the authorities where things can happen for you. And that way you'll be able to build the kind of thing that you saw in South Africa. I wish you luck and best wishes in the future. Thank you very much for coming forward.
Grateful. Thank you.
Thank you, Ma. Thank you, Ma. So we have a question in the chat box now.
In the message, or the presenter should?
No, okay, I will just read it for you. Sorry, I had a network glitch. So it says, how can we ensure the ethical use of human models? I mentioned about tailoring treatment to the patient's condition. How do we guarantee the solution reaches the patient in time before complications arise? And how viable will it be in real world situations? Yes, sir.
Okay, thank you very much.
Yes, sir.
This is a very powerful question because,
Mm-hmm.
well, one of the few things would be to probably use some alternatives that will likely give us a better result. The thing is, it is very difficult for us to predict what will happen in humans without actually using humans. So, and as I mentioned earlier, the results we see even in animals, and that's why extensive research in rodents, so to speak, is usually done before you move into human trials. However, it is still not robust enough because our biology is still not similar. So I think one of the few things, just as I mentioned, is probably getting these IPCS, that's inducible pluripotent stem cells. So if you can get humans that actually have these pain conditions, get their cells, induce them to express pain nerves or cells, and then begin to test on those cells. I think that is closer to humans than even using the patients. But with regards to the time, this is, in fact, from other development of medications, I think the average that I've had is 15 to even 20 years. But for pain, we said 10 to 15. So it's still within range. For now, I think maybe with technology too as well, that can also reduce that time significantly with AI and the rest of them. I hope this answers your question satisfactorily.
If I would say one or two things that there are ethical committees in the hospitals and in institutions. So every research that has to involve human being or animals actually pass through ethical review, which is very important. That whether it is on animals or on human being, there must be ethical review and approval. And so you have to weigh the benefits and the risk. And in some instances, I was listening to a lady who had to use experimental drug, and that was the only thing. And it was just coming from the laboratory, and there was nothing they could give to her. And she volunteered to say, look, whatever happened, I want to go for this test. And I have a professor of anesthesiology in San Diego, and she said she was having a cancer, and at that time there was no drug for that treatment, and it was experimental drug, and she volunteered to say, I'm going to take it. And she's still living about 50 years thereafter. So it's sometimes 50-50. But you just know that you have this ethical approval that cover whatever you are doing. And also, when we are talking about animals, I mean human, he has mentioned about cell lines and what have you. So it is not actually that you are using a whole human being, but you are using part of human being for that particular type of test. I have a very similar experience with him in postdoc activity because I did my postdoc in Department of Neurology, and I'm a basic scientist. And so my PI, who is a neurologist, he wanted me to model something, small fiber polyneuropathy in rats. And so he took me on to see patients when she was clerking a patient that had the same disease. And it was really, really impressive for me because real time I saw what I was going to model in rats, and it actually helped. So I think interdisciplinary nature and this kind of setup will be quite beneficial, as we have been talking about since morning.
All right. Thank you, Prof. Sir, for that addition. And thank you, Dr. Issa. We have a question, another question in the chat box, and he's saying that he's just been curious that has there been preclinical research involving animals with burn wounds, and is pain from burn wounds similar to the post-surgical pain that is under inflammatory pain? Dr. Issa.
Okay, thank you very much.
Yes, sir.
So interestingly, there are animal pain models for burn in humans. Some of them include the rat's hind paw bone, where the hind paw is immersed in about 85 degrees Celsius of water for 12 seconds. In addition to the previous questions, the ethics surrounding pain is actually very rigid and strict. In fact, if you read the Zimmerman on this ethics of pain, one of them is that in certain situations, if you can, you should actually be able to test even the pain model on yourself, and you don't harm before you use it on animals. And as much as possible, even when you carry out pain studies, you have to minimize pain as much as possible, which one should not even be heartless. So there are other ones too, the mouse-borne models, just a couple of them. It's just to find out which one is most suitable for you. Now, is burn pain or burn wound similar to post-surgical pain that's under inflammatory pain, so to speak? So inflammatory pain and post-surgical pain or burns pain, I don't think they have the same similar pathway, so to speak, because in burns, you basically have allodynia, not hyperalgesia, because you have non-noxious stimulus causing pain, and in this case, you have the free nerve endings that are actually stimulating it, unlike the post-surgical pain. So even though you still have the inflammatory soup or inflammatory mediators involved, but they are not completely similar because they have a different pathways, so to speak. I think I believe, I don't know if this is sufficient for you. Thank you very much.
Yes, thank you, sir. Another question here said, how reliable are rodent behavioral pain models in predicting subjective pain experiences in humans? It's a very important one.
Yes, as I read the comment section now, somebody said there's a lot of advances that have been made in the management of pain. Yes, you have the grievance model. So you still have those things. It may not be as good as the human study because they cannot self-report. That's where the limitation is. However, you can be able to assess if you understand neurobehavioral models. You know that you have some for assessing pain, and you can also assess for anxiety and depression too, which are comorbid, or they are also associated with pain as well. So that at least can give us some form of idea into this, but it is still not a perfect replication or representative for you having it in humans, but we are getting there. I doubt if it will be perfect. But
yeah, but let me just chip in here that just as we have said, we actually model these comorbid conditions, anxiety and what have you, and depression. And we have just like Professor Sogebi have said, and this is a vet, and of course they are experienced in this. I mean, these days now, it is not just to test the pain itself, or sometimes you have a design whereby you actually measure the depression and what have you, locomotion and all the behavioral characteristic associated with the pain perception. So it is more now than nociception, but it's tending toward pain. And somebody was saying how the commonest one that we talk about, hot plate. There were some of these models that were used to determine that aspirin is good, opioids are good. So
Yes.
there have been some of these basic model in the laboratory in animals have actually predicted some of the drugs that we're actually using today.
Yes. And they've been successful.
I think somebody is raising up the hand, moderator. So
Okay. Okay.
We must also remember that you don't have you may not have unitary kind of pain.
Yes.
And also the fact that you can be dealing with acute pain or chronic pain
Yes.
and a mixed kind of pain and
Yes.
complex pain, as he mentioned in his
Yes.
presentation.
Okay.
thank you, ma.
So that is why it's important for us all to work together, because eventually we are treating human beings or animals. And the people who are treating them are not necessarily those ones who are performing all the research. There are scientists who are pure scientists, but by the time you talk to them, you will think they are clinicians. Thank you very much.
Thank you, ma.
Thank you, ma'am. Yes.
Seun, can you
Seun.
unmute yourself and talk?
Yeah, thank you so much. Doctor, thank you for the wonderful presentation. I have a couple of questions. The first one, do you think we would get to a stage where we would have like a brain biomarker or like physiological biomarker for pain, and is able to stratify the pain into probably different, say, grade, like grade 1 means like a very bad pain, and grade 5 means like very acute or manageable pain. Do you think we are going to get to that level? And the second question is, other than the translatability of preclinical data into human, what do you think is the most challenging issue facing the pain research?
Okay, is that all? Right. So to begin with, whether we'll have a biomarker that will probably indicate, if I understand you correctly, the type of pain, not the severity of pain. Is that right? Sure.
Yeah, more like type, maybe chronic, acute, or more like maybe severity, so that pain are kind of like stratified, and then that kind of make the clinician to know, okay, this is the extent of pain, medication I could give this particular patient, and then it remove the subjectiveness of pain, because the way
Yeah.
I talk about my pain might be different from the other individual, from the other patient, but how can we introduce objectiveness in like pain research?
Yes. Okay. Well, that will be an utopia for pain research if we can actually get that. And I've also thought about even just having a biomarker that once you measure it and the level of concentration will indicate the amount of pain that you have. However, pain is very complex. There are so many factors that affect pain. The food you eat, the culture you have, the experiences you've had previously. So many things have this interplay in coming out as pain. That's the final output. So just having a biomarker that will be able to summarize the whole of this. Personally, I think it's going to be difficult, very, very difficult. I don't think, well, perhaps we'll get something close, but we'll probably not get something perfect. That is what I feel. And with regards to the major challenge, I think Professor Sorewo just mentioned it. One of the major challenge in this translational thing, apart from that, is the fact that people experience different forms of pain from different origin, different mechanisms. There are so many overlaps. It's not just one plastic pain mixed various mechanism of action. And I think that is one of the major. You don't have this homogeneous collection of pain. It's a wide range, and then it's highly heterogeneous. So that is one of the major things I think, apart from the translational part, that I think affects pain. So it's not one size fits all. You cannot just say, and you actually give, if you give pain medications to a particular patient, it's also possible that if you give to another person, it doesn't have the same relief until you probably look for something that is suitable for the person. And that was why I said even the management of pain should be based on probably mechanism that you understand on what the person is having. That is my own take on that question. Thank you very much.
Yeah, thank you, Dr. Isa. Thank you. We have one more question in the chat box. So before we call it a night, he said, how does preclinical evidence influence multimodal pain therapy approaches in clinical settings? And how can advancements in AI and computational modeling improve translation?
How can preclinical evidence influence multimodal pain therapy? So it's one of those things that I just mentioned, because preclinical pain research, one of the major things it does, apart from identifying the target, is also making us understand mechanism of action. And if in certain situations you discover that in certain pain conditions, there's not just one mechanism of action that is involved. You have a myriad or plethora of things that are involved at the same time. Take, for instance, you are talking about pain that involves oxidative stress as well as inflammation. And you also have nerve damage too. So all these work hand in hand, and oxidative stress and inflammation is the situation of the chicken and the egg. So you probably want to tackle it from both sides, because you don't know which is driving which. So I think that will serve as evidence for multimodal pain therapy. With the role of advancements in AI and computational modeling, as I mentioned earlier, AI can actually be used to gather data from real-time patients, as well as using computational modeling. Because this helps to reduce or probably shorten the time that we spend in doing certain things, and so that we can actually be able to get better results. So using AI, in fact, you can actually do a lot of things, interaction of bioinformatics and the rest of them, to try to see the interaction between certain receptors and what effect it will have, and begin to even predict what you are likely to see even before you begin to go. I think it's something that hazard and call it the dry lab. Instead of going into the lab, you can actually model these things and run simulations rather than having the animals. So this can significantly improve our outputs and the translation from preclinical to clinical pain research. Thank you.
Yeah. Thank you so much, Dr. Isa, for answering our questions beautifully, and I think it gave us a well-rounded perspective as well. So I would once again want to appreciate everyone for joining us and for staying true till the end. As we wrap up, I would give the floor to the president, Professor Oyeli, to say one or two things before we close.
Thank you, Ms. Evelyn, and thank you for handling the moderation effectively. We want to thank our presenter, Dr. Sharif, for a wonderful presentation, and then the answers to the question, and then presenting all the problems and then the solutions that are just ahead of us. And I think this will help us and let us understand the importance of the interdisciplinary nature that the society is and it should be. So we want to appreciate Dr. Sharif, and I want to also thank Mama Sonyawo, the foundation president, for always standing with us and by us, and for the insightful comments that she has given this evening. I know that she has always been in favor of transdisciplinary. Now we can see how important it is when we work together. Like you see, Professor Sogebi coming from the angle of a veterinarian, and then we are coming from, Dr. Sharif is coming from a basic scientist, and yet we still deal with animals, but there are a bit of differences. and then we now move to human beings, the patients. And I know there is still a lot of opportunities for us, even in the country, because, for instance, one is that the Fulani don't feel pain the way the Igbo will feel pain, neither, and the Yorubas don't. That was one of my one research that I carried out, and there are differences, both genetics and both constitutional wise chemicals that are present. So, and I think the good understanding of this is very important for us in moving the treatment and understanding of pain forward in the country. So I want to thank every one of us. I want to thank Mama. I want to thank my immediate President, Professor Akinpelu, the members of the executive, and everybody that have found time to attend this webinar. I want to say that the webinar is recorded, and as usual, we will post it on our YouTube channel, and people will have access to look at it later. And so if we have questions, we can still meet the presenter or ask them on our WhatsApp channel. And we want to encourage everyone, if you are here, you have attended this webinar and you are not a member of the SSPN, please try to join. From wherever you have gotten this information, you can ask for how to join the SSPN. Is the most diverse group that is involved in the study of pain, both locally and internationally, and it's an interesting group to join. So we thank every one of you and wish you a good night rest. All right, so the secretary have put his number on the chat box, so you can reach out to the secretary for how to become a member. And thank you very much. So I don't think we have any other thing to do than to thank you, thank Dr. Serif, thank the moderator, thank everybody, and say good night again.
Thank you very much. Good night, everybody.
Thank you very much.
Thank you, Dr. Sharif.
Good night,
Thank you.
ma.
Thank
Good
you.
night.
Good night.
Yeah, thank you, Dr. Sharif. Good night.